Okay. As you know, we've already started this whole section on what is truth. We've already gone through the first 5. Number 5 there was when we talked about the authenticity of the Scriptures. This is the one we're on now. Number 6, truth does not equal science, and science does not equal truth. And I've been setting this up the last couple weeks. Now, from this point on, and like I said, depending on how long this goes, we're going to start talking about some data. And we're going to start off focusing on DNA. And the reason is because I told you last week, that's my area of expertise. And this is the center of this entire issue. Because if you don't have DNA, you don't have life. All right? So if you can't explain the origin of DNA, then you can't explain the origin of life. That's why it's the central issue of this supposed dilemma. And not only can evolution not explain the origin of DNA, but its structure, its functions, and its very existence clearly show us that evolution is not possible scientifically. And please remember, the fundamental aspect of the theory of evolution is that life came from non-life. Okay? After the supposed Big Bang that happened supposedly 20 billion years ago, and then about 4 to 5 billion years ago, our Earth formed, according to this theory. Well, we can stop right there because I already mentioned this, but let me just repeat. The glaring issue that comes up right there. And the obvious question you should be thinking is, where did the extremely dense and compressed energy and matter come from that supposedly exploded in the Big Bang? See, there's issues all over the place. That should be the first question right off the bat. Okay, but anyway, back to their theory. Then supposedly around 1 billion years ago, all the molecules necessary for life, your DNA, your RNA, your proteins, both formed and assembled all by themselves as a result of the presence of the right atmospheric and environmental conditions. Then the first cell formed and developed all by itself. And life has been evolving from that first cell ever since as a result of genetic mutations. So in a nutshell, that's their theory. And if you see what happens here, according to their theory, DNA had to have arisen spontaneously, all by itself, as a result of randomness. There is absolutely no way around this for evolution. None. That's what had to have happened. And also, like I mentioned with the thing with the Big Bang, keep in mind, it's the origin of matter and life that's the most critical, yet it's the one aspect of this supposed controversy that evolution has absolutely no answers for. Evolution has no answers for where the energy and matter came from, and no answers for how life could have come from non-life. None. Please remember that. In the book Charles Darwin wrote, The Origin of Species, the greatest irony is that the title says the origin of the species. However, his book says absolutely nothing about the actual origins of life. Keep that in mind too. And the reason why that is... Oh, sorry, I missed one. My bad. The reason why that is, why it says nothing about that, is because nothing in science, nothing, has ever been able to explain how a molecule like DNA, which life depends on and is based on, could have come about through random natural processes. And the reason why that is because, folks, it's not possible scientifically. And that's what I'm going to share with you over the next how many weeks. You see, DNA is so amazing, and it really is, and we're going to take a look at that through its structure and its functions, it tells us about its origin. But before you can understand that, you have to first understand the structure of DNA and its functions. So that's what I'm going to spend some time doing now. And in doing that, let's see, do I have... Okay, here we go. So I mentioned DNA. First of all, anybody know what that stands for? Somebody's got to know what that stands for? No? Deoxyribonucleic acid. That's what DNA stands for. And it gets that name from its structure. I know you guys know, you just weren't paying attention.
We're trying to figure out the
Yeah. It gets that name from its structure. So I want to go through that now, okay? First of all, you have to understand DNA is unlike anything else in life. DNA is truly unique. But it's not only unique, it's complex, it's organized, it's choreographed, it's controlled, it's structured, it's patterned, it's arranged, and yet it is so simple. It's a biomolecule that stores information. And not only that, it's a massive amount of information that is incredibly complex, choreographed, organized, patterned, and logical. And yet, it has only 4 building blocks. This is truly amazing. But it not only stores information, it utilizes information to create, develop, grow, sustain, reproduce, protect, and heal life itself. DNA contains the information needed to recognize and repair damage and errors, to reproduce itself, to regulate itself, to protect itself, to store itself, to turn itself off and on, to not only make proteins, but to know which proteins need to be made and how much of them. To reproduce life itself. I could do this all day with all the things you can do, but hopefully you get the point. The bottom line is, without DNA, life as we know it would be impossible. Therefore, evolution would have to account for not only the origin of DNA itself, but also the origin of the mind-boggling amount of information that it contains and utilizes for all of its life essential functions. Any attempt to explain the origin of life absolutely must include these 2 things, both where the structure comes from and where the information it contains comes from. This is non-negotiable no matter what you're talking about, okay? And here's the problem evolution has. It has no answers for any of this. None. Evolution cannot explain a biomolecule that contains and processes information in a manner that is far superior to anything man has ever created or ever will create. Nothing in mainstream science has ever been able to explain this. Here, let me give you a quote. I went too far. There we go. Dr. Werner Gitt states the following. There is no known law of nature, no known process, and no known sequence of events which can cause information to originate by itself in matter. That's a problem for evolution. The simple fact is that evolution never has and never will be able to explain DNA or the information that it contains. Therefore, evolution cannot explain the origin of life. And it actually is that simple. We could end our discussion right there. We really could. Because just because of that, evolution has already lost. But if you doubt me, take a listen to what the evolutionists themselves say. This following quote is a summary from the 4th Congress of the International Society for the Study of the Origin of Life. And in this is their summary statement. For the first time, it has now been determined, unequivocally, by a large number of scientists, that all evolutionary theses that living systems develop from polynucleotides, which originated spontaneously, are devoid of any empirical base. To make sure you understand what he's saying here. He's saying a large number of evolutionists have jointly determined that there is no data or scientific evidence to support that DNA and life itself could have originated spontaneously, straight from the horse's mouth. This is both an extremely eye-opening and damning statement from the evolutionists themselves. In this one statement, they have admitted defeat and admitted that their theory has no leg on which to stand. Together, they've decided what we already knew. There's no evidence, no experimental data, no observations, no practical reason, and no explanation to support the origin and existence of DNA and its information coming about through evolution. So like I said, we could end our discussion right here with just this one point. And please get this. The evolutionists themselves know it too. They have admitted it, and that's not the only quote that you can find like that. You'll be seeing more. That's why they have never, ever come up with a plausible explanation for the existence of DNA or the information it contains. Nor will they ever come up with one, because it's simply not possible scientifically. So what they do instead, they rely on all their rhetoric. They have what I like to say, they perform their two-step, right? They dance around it because they have no explanation. And in doing their two-step, they use big, intelligent-sounding words, right? To make you think they're so smart that I should just believe what they say because they got to know what they're talking about. So what they do is they rely on avoidance, detection or deflection, sleight of hand, you might call it, misappropriation of data, deception, and as I'll show you, outright lies, because they simply cannot rely on science or data, because it is not there. The science and data destroy their theory, so they can't rely on it. The DNA molecule itself kills their theory. Because like I said, you can't explain it via evolution. And like I already shared with you, they know it. So I encourage you to do this. The next time you encounter someone who believes in evolution, ask them to explain the origin of DNA to you and the information that it contains. Go ahead and ask them that, and then sit back and enjoy the show, because that's all you're going to get from that point on. Now, I said I could end my discussion here, but that would take all the fun out of it, right? I would thoroughly enjoy beating the dead horse of evolution. So, like I said, we're going to do this for a while, So we're going to begin today moving on, talking more about the structure of DNA. DNA is made up of only 4 building blocks, like I said. The 4 nucleotides, each of which are composed of a nitrogenous base, that just means it has nitrogen in it, a deoxyribose sugar, and a phosphate group. And I'm going to show you some pictures of this. All the chemical bonds, this is important to note, all the chemical bonds in a nucleotide are covalent. And we're going to talk about this because this is critical. A covalent bond is one where there's an equal sharing of electrons between the 2 atoms. So you end up with a very stable interaction between 2 atoms. So covalent bonds end up being pretty strong. So if you look at a structure like, let's say, a car or a building or something, you could say the covalent bonds are like the welds in the steel used, or they're like the nuts and bolts, or the rivets, or if you're using wood, they could be the wood screws or the nails. That's what holds it all together, and if you remove those, the whole thing falls apart. So that is an analogy of what a covalent bond is. When you look at this structure, here's what it looks like. Here's our 3 groups. Here's our base, our sugar, our phosphate group. In a 2D image, this is what it looks like. Each of these are atoms. Oxygen, hydrogen, carbon, phosphorus, nitrogen. So this is how they're arranged if you look at it in a 2D image. And like I said, the 3 different groups are part of the nucleotide. One other thing to point out on this. A lot of times when they draw these structures, the angles, there's actually a carbon atom in there, but they don't label it for simplicity of drawing. When we look at the ribose sugar part of it, all of those carbons are numbered. You see we have 1, 2, 3, 4, and then 5 up here, and they get a little prime on the end of them. So it's 1, 2, 3, 4, and 5 prime. I'm pointing that out to you because this is going to come up later. This is actually very important. And one more thing to point out. This is where DNA gets its name, right here. In a typical ribose sugar, there's an OH here and an OH here on 2 and 3 prime. However, in DNA, the 2' position is missing the oxygen. So it's deoxy. Therefore, it gets its name from that. Deoxyribose, or deoxyribonucleic acid. So that's where its name comes from, right there. I won't charge any extra for that. I'll throw that in free of charge, okay? In the base region, the one that was up here in the corner, there are 4 different bases that can fill in that region. These are the chemical structures of those. Notice these positions get numbered 2. However, they don't have a prime. That way you can know whether you're talking about a carbon in the base or a carbon in the sugar, whether or not it has the prime. And like I said, there's a reason I'm pointing that out. But here are the 4 different bases that make up the 4 building blocks of DNA. Adenine, guanine, thymine, cytosine, or what most people just refer to as A, G, T, and C. They usually just use the first letter. So it usually breaks down to saying the 4 bases, 4 nucleotides, they call them by their base and just call them A, G, T, or C. It's a lot simpler than trying to repeat all that mouthful all the time, right? Okay? So let's move on. By changing this part right here, the nitrogenous base, so by swapping that out with one of those 4 structures we looked at, that changes the nucleotide into being either an A, G, T, or C. So when I started saying 4 building blocks, That's the 4 building blocks of DNA, A, G, T, or C. Now, I said the nucleotides are held together, all the atoms, covalently. When the nucleotides are added together to each other, they are also held together by covalent bonds. And I brought some toys today. Legos are a great way to demonstrate this. If we imagine this being one of our 4 nucleotides, when DNA is created, another nucleotide gets attached to it. And it's covalently bound, so on and so forth. Just keeps going. These can be added one after another, millions or billions of times, depending on the size of the genome. And I'm actually going to give you some examples of different animals and the different sizes of their genomes. When they do that, I was showing this for simplicity reason of how they attach to one another. But this is really what a nucleotide would look like, right? Phosphate group, sugar group, base. The 3 parts. The phosphate and the sugar are more in line. The base kind of sticks out into the middle, kind of perpendicular to the phosphate and sugar group. So it would look like this. So if you took nucleotides, put them together into long strands, they'd look like this. Big long strand of all these, where our backbone region, sugar phosphate, with the bases sticking out perpendicular from that. So it would look kind of like this, okay? Let's see, let me go on here. Then what happens, I'll take this apart because this is all I have. 2 strands, 2 separate strands. Let's imagine this one being really long and this one being really long. Get brought together. Actually, it would be like this, but they won't stay together like that. They get brought together like this. Where the backbones are running up and down, the bases are sticking out in the middle across from one another. And when this happens, there is a pattern to how they go together. There's 4 different bases, A, T, G, and C. A always binds with T. G always binds with C. So you find an A on one side, you're going to find a T on the other. A on this side, T on the other. G on one side, C on the other. G on one side, C on the other. They always go together, A with T, G with C. When we end up doing this, what does that end up looking like? A ladder. It ends up looking just like a ladder, doesn't it? And, let's see what we got here. There it is. Look at that. Talk about a segue, huh? DNA is often compared to being analogous to the shape of a ladder because of that. The paired bases in the middle form the rungs, right? The sugar and phosphate on the sides form the sides of a ladder, the structural supports. So DNA ends up looking a lot like a ladder. However, it doesn't just end up like this straight. A good way to show this, if you take a rubber band, now you have to imagine the bases are in the middle, but if we do this with the rubber band, this is what it's actually like. That ladder then gets twisted. Corkscrew, spiral staircase pattern, whatever you want to call it, where the bases are still in the middle, but it actually ends up looking like this. This is why DNA is referred to as a double helix, because it's shaped like that. Okay? Let's see what we got next. Here is the important part, what we're getting at here. The bases in the middle. These guys in the middle. They get held together by hydrogen bonds, which are different from covalent bonds, okay? So DNA is basically two covalently bonded molecules held together in the middle through the bases by hydrogen bonds. Okay? Here's what that looks like in a 2D image. Along the sides, sugar phosphate, sugar phosphate. Same thing over there. C binding with G, T binding with A. Hydrogen bonds holding them together. Everything else is covalently bonded. A always with T, G always with C. If you looked at an image of a 2D image of what DNA looks like, that's what it looks like right there. Here's another thing I want you to notice why I talked about the primes. Look over here on this side. If you follow 5' to 3', it's going that way. If you follow this one, 5' to 3', it's going that way. So not only is DNA two covalently bonded molecules held together by hydrogen bonds, they're two covalently bound molecules running in opposite directions of one another. That's why I was pointing out the primes. Because of that, you can see that each strand runs the opposite direction of the other. Okay? And all that to point out, DNA, on top of everything else, it has directionality. And this becomes very important when we start talking about some of its functions. So they're not only just two covalently bonded molecules held together in the middle. Like I said, they're going in opposite direction. And this becomes important because you can only add nucleotides to one of those strands in one direction. We go back to our Legos. You can't put Legos together like this. You can't put them together like this. They only go together like that. Nucleotides are the exact same way. They only go on in one direction. Just that one strand is going, as in this example, up. The other strand, it would be facing down like this. Okay? Now I said there can be millions to billions of these nucleotides in organisms' DNA. Let me give you some examples of that. Oh, sorry, I'm getting ahead of myself. Slow down here. In the DNA of an organism, in the DNA of an organism, there we go. People refer to its collective complete set of DNA and genes as a genome. But in higher organisms, that is often broken down into shorter segments, which we refer to as chromosomes. That's typically in a higher organism, I can't say organisms. Critters. Should we go with critters? Higher organisms, like us, for example, we have chromosomes. However, what people usually refer to as lower forms of life, the lowlifes, the bacteria, for example, they do not have chromosomes. Their DNA is all in one long strand. Okay? Chromosomes also come in pairs.
Is their DNA double-stranded or just single?
It is also double-stranded. Oh, there it goes. Chromosomes come in pairs. So in talking humans, for example, we all have a pair of every one of our chromosomes. So we have 23 pairs of chromosomes. The reason that is because you get one set from your father, one set from your mother. And those two sets are not identical. There are minor differences between the two sets. And a good way to say that is you get all the same genes from your parents, but not all the same versions of those genes. And a good example for that is you could say one gene version, or what is called an allele, that you get from your mother might be for brown hair. The other version or allele that you get for your hair from your father might be for red hair. They're both to make your hair. However, they're not exactly the same because one could be for a different color. Yes, sir?
Okay, I have a question. Is it possible to have like a trait that you get from your parents that doesn't show, but then it gets passed down to your kid, but it's
Absolutely.
the word that I'm looking for, like
Recessive versus dominant.
yeah, like it's recessive in you, like you still have it, but it
Happens all the time.
Yeah.
And that gets into a whole bunch of other genetics, and maybe we can talk about some other time, but it happens all the time. People can be carriers of a trait that isn't expressed for several different reasons, one of which could be it's a recessive trait. But that happens all the time. Okay? So this is what's referred to as genetic variation. Not every gene is in the same version. And this is important because this is a huge area of deception for the evolutionists, which is why later on we're going to talk more about that. But I was telling you that those nucleotides can be linked millions to billions of times, depending on the animal. I'll say animal instead of organism. There we go. Here are some examples. E. coli, a bacteria, has 4 times 10 to the 6, which is 4 million base pairs, all in one molecule. Chimps, 3.35 times 10 to the 9th, which is 3.35 billion base pairs, and they have 24 pairs of chromosomes. Mice, 2.5 billion base pairs. They have 20 pairs of chromosomes. Dogs, 2.4 billion base pairs, 39 pairs of chromosomes. And like I said, we have 3.2 billion base pairs divided into our 23 pairs of chromosomes. So you can see it varies a lot depending on the organism. Yes, sir?
I feel like 23 is the sweet spot. Like if you have like 24 chromosomes, which is like mentally handicapped, so if you have less, why is 23 like?
Because each organism has the amount that is unique to that organism.
So if the dog had more than its chromosome?
But it's natural and normal for a dog to have that many. Humans, it's natural, normal for us to have 23 pairs. So it's only when you get outside the normal number that you start having trouble. But for each organism, that's what I'm showing you, they have a unique set of number of chromosomes.
Why does the dog have
Is there an advantage or a disadvantage to the number?
Not like
No, not from organism to organism. It's not really that. It's just that is the way the amount of genes are required in that organism.
To create.
That's correct. That's correct. Okay. So, I just said we have 3.2 billion base pairs divided into 23 pairs of chromosomes. Let's show you something amazing here. If you do the math, it comes out that humans have about 6.5 feet of DNA in each cell. So one cell has about 6.5 feet of DNA if you pull it and stretch it end to end. And adults have about 50 to 100 trillion cells. So again, doing the math, if you put all the DNA from us end to end, you end up with at least 61.5 billion miles of DNA. One person. Now, to help you understand how truly amazing this is, we have enough DNA in our bodies, cumulative, to stretch from here to the moon 258,000 times. To stretch from here to the sun 660 times. You see what we're getting at with all this, how amazing DNA is, right? But if that's not enough, take a look at this. All that from one cell. Start looking at just one cell, 6.5 feet long, weighs only 3.27 picograms. That's 10 to the minus 12 grams, or 0.000000000327 grams. I had to point that and make sure I got all the zeros in there. Now, if we look at the entire amount of DNA from a human, 61.5 billion miles, to the moon 258,000 times, to the sun 660 times, it weighs only about 163 grams. Folks, that's about a third of a pound. Do you see how amazing this is? Hopefully you're grasping that. But that's still not the end. All that has to fit inside a cell. We have to get 6.5 feet of DNA inside a cell. In order to overcome this storage dilemma, DNA undergoes extensive levels of packaging and condensing. And this involves proteins called histones. And histones, because of the way they're made, they have an affinity for things with negative charges on them, which is exactly what DNA has. So it has an affinity for DNA, meaning it's attracted to it. So DNA wraps around these proteins like beads on a string. You could say it that way. And then that undergoes coiling into what's called a 30 nanometer fiber. And that gives DNA a rope-like appearance. And then you have more proteins, scaffold proteins, that then twist that up into wave-like structures. Those are called 700 nanometer structures. Do you see where we're going with this? You end up with an exceedingly structured, ordered, supercoiled, supercondensed pattern of folding that DNA has to go through just to fit inside a cell. So on top of everything else, in order for DNA to function, before you can do anything with it, you got to get through all that. You got to unwrap it, get through all that condensing and folding. And this is important because the life essential information from DNA is in the middle of the molecule, where the base pairs are. The rungs of our ladder that are hydrogen bonded to one another, that's where we find the information that you have to get through. So you have to get through all that supercoiling and get inside the DNA molecule for life to exist. And folks, what I just shared with you today, I've only scratched the surface. We could do this, although there are entire courses on this. We could do this a long time discussing the intricacies of the structure of DNA. But what I'm hoping you're getting from this, it's truly amazing and unique beyond our wildest imagination. And looking at the structure, the patterns, the uniqueness is in itself enough to convince any objective thinking person that it's not possible for it to come from randomness. But there are, with that said, there are specific points about the structure. Now that I've shared this with you, I'm going to go through and point out why it's important. Most of that we're going to do next week. There's just one that I'm going to point out today. The bonds. The types of bonds and the specific position of the bonds. Hydrogen bonds are used in the middle to hold the bases to one another. They hold the rungs together in the middle. They're hydrogen bonds. All the rest of this is covalently bonded. Okay? That is really important. Because the hydrogen bonds are weaker bonds. The covalent bonds holding this together are all stronger. The hydrogen bonds are weaker bonds. Hydrogen bonds can be broken and reestablished without destroying the integrity of the molecule. That's not so with covalent bonds. If you break covalent bonds, you will destroy the integrity of the molecule. Okay? So the presence of hydrogen bonds in the middle enables the two halves of DNA to be split open, have the information accessed, close back up again, all without destroying the integrity of the molecule. So what ends up happening is you can open and close DNA just like a zipper. That's the best analogy for it. Just like a zipper. Like a zipper. You can zip it open. The bases would be like the teeth in a zipper. You can pull the clamp down and separate them. Pull the clamp up, bring them back together. In doing so, we're not destroying the zipper. Just open it and close it. But if you took the stitches out of the zipper, then it falls apart. So the stitches become like the covalent bonds. They have to be there or it falls apart. The hydrogen bonds are like the clamp running up and down, opening it and then closing it again. I say all that to say life depends on this structure. Life depends on the ability of DNA to be opened up, have its information accessed, closed back up again, without being changed or altered. That is absolutely critical. It has to always remain intact and unchanged. If it were to change all the time, or if it was to get destroyed, its reproducibility is lost, its ability to do its functions is lost, all of which is mandatory for life, and therefore life itself is lost. So life then ends up depending on the correct placement, order, choreography, if you will, or pattern of each hydrogen and covalent bond found in the DNA molecule. And that also depends on the placement of each atom, because not all atoms can form hydrogen bonds. They have to be the ones that can form it, have to be in the right spot in order for that to happen. We're going to talk more about this next week, actually. So, this cannot be left to randomness. You have to know the difference between those two types of bonds. You have to know that. You have to know where you can use the covalents, where you can use the hydrogens. If not, if they're all randomly in there, DNA will not be the molecule that it is. And like I said, life cannot exist because of that. So you have to know that. It cannot be left to chance or randomness, or it will not turn out the way that it is. I basically said that. You have to know where a hydrogen bond will work and that a covalent bond won't work there. You have to know where a covalent bond will work and that a hydrogen bond won't work there. If left to randomness, you do not get that. Randomness will not create the specific life essential pattern that we find in the bond usage in DNA. Randomness will create bond usage that is random. Thank you. I was waiting for somebody to say that. Random because it's random. Thank you. If left to randomness, the bonds will be random, DNA won't be what it is, DNA won't function, life could not exist. So just by looking at the pattern of bonds, just by looking at that all by itself, we learn DNA could not have come about as a result of randomness or chance. And that's just looking at the bonds. There is much, much more that we'll be talking about over the next couple weeks. And that's where we're going to end it today. But I have a lot more to share with you over the next several weeks.